Okay, so we can start. No, you guys have next week, Jorge Carneiro. Yes. So it's interesting because he's going to be kind of complement the immunology with a bit of modeling mathematics, I think. That's what he does, what I remember. I once spent a few months in the Gulbenkian next to his lab. So, but is that what he does? Immunology modeling. Yeah, but I think it has immunology component and has modeling component. At least used to have. Okay. So we were supposed to have a conversation with about any topics. I think the one topic that was raised that apparently was the consensus was kind of career paths. And then there are a few other suggestions that we said that we're going to go as we discuss, right? Any other suggestion or any other, do you guys want something different or it's okay? For example, do you want to continue with the kind of research topic presentation that I was doing or you want a discussion? Yeah. Okay, so what I can do is to present the final part of what I was supposed to present. It were 3 parts, and then after that we discuss. Is that okay? That's fine too. Do you guys know how to change this? No. It's not connecting. Is it? Okay, so I'm going to just complete what I was presenting, and then maybe half an hour or so, and then we discuss the rest of the time, the kind of discussion topic. So what I didn't present, I just want to complete this second part, because otherwise it was just a historical thing. So one of the things that the lab was interested in the topic of tolerance, conventional immunological tolerance, was how can the intestine deal with, and I was discussing a little bit when we discussed the lipstick and the dendritic cells stuff at the board here. how can the intestine deal with infections and absorption of nutrients at the same time? So, the mechanistic aspects I discussed here, but in terms of data, one of the things that I wanted to complete that we found that we thought was very interesting is that the gut, very long, has too many. In humans it's even more, but in mice there are a number, maybe 12 or 13 gut draining lymph nodes. So the lymph nodes, of course, is where the initiation of the immune response takes place. Everything is drained there, both solubly as well as carried by the dendritic cells that migrate from the tissue. And what was surprising, is actually that was interesting because this postdoc, Daria Esterhazy, she had expertise in metabolism, and she kind of was wondering, and I think that was a question of someone who came from outside of the field, whether the gut draining lymph nodes that are draining different regions would be very different from each other or would be similar from each other. And what we found is that in terms of cells that are induced to mediate tolerance, the draining lymph nodes of the proximal part of the intestine that drain the duodenum are specifically strong in inducing these cells that express FOXP3. These are regulatory T cells. So it was kind of, so maybe the tolerance in part is mediated because the proximal draining lymph nodes are more tolerogenic than the ones that drain the distal parts of the intestine. These regions here, for example, is where most of the infections, invasive pathogens, and so on take place. So there is an anatomical, what we call compartmentalization of the gut immune responses by where they are draining from. And how this is induced, we also addressed this in this story. For example, the long-chain fatty acids are absorbed here. There is a concentration of retinoic acid pathway, vitamin A metabolite in these regions. These all make these lymph nodes quite different than the lymph nodes that are draining the distal part of the intestine, the ileum, and the colon. Okay? And we did a lot of imaging on this. It's not functional. It's just interesting image. So this is kind of, we were doing this early on. But this is, basically, it's interesting. This is called iDisco. So you make the tissue clear so you can image the entire tissue in one block. This would be difficult in humans, of course, but you can do in humans as well. We have done. So here is each villi is is the intestine, see, the duodenum. And in green is the pancreas, because this is stained with insulin. You have liver, and you have these blocks here, which are the gut draining lymph nodes. And what you see in white are the lymphatics. So the gut has these lymphatics, and the lymphatics are carrying antigens and cells from the gut draining lymph nodes all the way to the gut tissue, all the way to the gut draining lymph nodes, which are compartmentalized. And then this is just to show how the segregation occurs. And we just kind of went on and characterized the mechanisms of this differences. I'm not going to go in details on this, but basically what we found is that these initial lymph nodes here, which are draining the proximal part of the gut, they are very different and they are more prone to induce tolerance. This is kind of how the lymphatics structure, again, no functional here, how the lymphatic structures are organized in the gut. So each one of these structures, again, is one villus. We covered a lot of villus in this class. And each one of these is a lacteal. A lacteal is the afferent lymphatics, the lymphatics that carry stuff from the tissue to the draining lymph nodes are organized. So these are collecting vessels. You see they extend all the way to the tip of the villi, and then they kind of end up in these larger vessels, and then the larger vessels, and then they go and migrate. See, these are lacteals of each one lacteal per villus. And then they carry the stuff that's present in the lumen, cells, and so on, and they go in the larger lymphatics, and then this will end up in the gut draining lymph node, basically. Okay, and these are each one of them. But anyway, I'm not going to spend too much time on this.
Yep. Can you just go back?
Yeah.
Yeah, one more and one more. This, no, the other. The next one.
Yeah, I'm going. It's just slow.
Yeah, so then this image is kind of counterintuitive for me because then you have this gradient of the microbiome that is decreasing
Yeah.
when you go through the intestine, but then like the biggest concentration of nutrients will be in the first part, so why this this direction of this gradient?
Which gradient?
Of the microbiome.
Why is a teleological question.
Yeah.
I can't answer. But what I can say is that the conditions for the bacteria to survive and proliferate, it's better and more optimal in the distal, in the distal gut. So for example, in the way that the stomach and enzymes are organized in the proximal gut, it makes less prone. Only lactobacillus can survive in the proximal small intestine. Then you have a regulation of the bile, bile acids and so on that mediate sporulation. For example, these are a lot of kind of physiology stuff, but in the cecum, for example, you have, that's where the bile acids will end up. And bile acids are known to take a spore-forming bacteria and kind of sporulate and then make into bacteria. So, and this kind of process is back and forth because the bacteria that is formed in the cecum and proximal column also can convert primary bile acids in secondary, which are then fed into. So there's a bunch of stuff happening in the cecum, which is this proximal part of the large intestine, that makes I would say a more suitable place for bacteria species to survive. I mean, there are probably many more. For example, mucus layers are deeper, are thicker in the large intestine. But, so there are many reasons for that. Genetically, there's actually interesting, the first paper that I know of that explained why the duodenum is so different from the ileum is a paper from Bana Jabri. I don't know even if it's published, as long as it's in bioarchives, but it's a transcription factor GATA4 that if you knock out, the ileum looks like the duodenum or vice versa. I forgot what's the direction. But there's some genetic imprinted developmental stuff that make, for example, the ileum, the villi is very short. In the duodenum, the villi is very long. In the large intestine, there is no villi. It's only crypt. So there are anatomical differences, physiological differences like enzymes and so on, that make this place kind of, in a way, better. There are about 1,000 times more bacteria in the proximal colon than in duodenum, for example. In the number of species also increase. So, there's a long answer, but there's a lot of these explanations, why you have more. But if you're asking why you have more or less immune system to the or what you ask is what I just said.
Basically what you said.
Yeah.
I was thinking that the part that would be more colonized was the first one because it would be the ones that has the major part of the nutrients.
Yeah, that's true.
And not be absorbed yet.
Yeah.
But then
on the other hand, the nutrients in the proximal part are more intact. So the nutrients availability actually for bacteria or what they need, for example, fibers and so on, increase more distally, while the kind of intact proteins and lipids indeed absorbed there. But then you have different kinds of nutrients availability for bacteria. Mucus, for example, a lot of them feed on mucus and increase in the distal intestine. But what is surprising, perhaps, and that is not answered, is the concentration of the immune system doesn't really match, depending on what you call immune system, but amount of intraepithelial lymphocytes, for example, it changes in a way that is not predictable based on bacteria. Because, of course, what happens in the large intestine is that not only you have more bacteria, but you have is less absorptive. So you have kind of barriers instead of immune system only. Anyway, it's a lot of stuff that's interesting in this kind of interplay. But what we try to understand is how the immune system is very different in the proximal part that's dealing with nutrients versus the distal part that is dealing with the microbiome. That's kind of part of this question here. Okay. So the last part of this is an example of disease tolerance that we talked a lot. And this goes to the area of the lab, the third area of the lab, which is neuroimmune interactions. And I began my interest in this since my undergrad in the lab of Nelson Vaz in Minas Gerais. Because Nelson was studying something called food avoidance behavior. And it's something that's very interesting. I'm not going to cover here, but how allergic mice know to avoid the allergen. It's kind of a very interesting kind of neuroimmune intersection that we are actually studying now as well. But the main reason that we started this is this picture here. This is another eye disco, like clearing of the intestinal tissue. And these are the neurons in the gut. And these are both the neurons that live within the wall of the intestine. These are called intrinsic neurons. And these are also known as the enteric nervous system. And also these neurons that project fibers to outside the gut. That includes fibers that are sensory, so sensory fibers that are sensing whatever, this tension of the intestine, content, and so on, as well as projecting fibers that are projecting from the gut to outside. There is sympathetic fibers that are coming from sympathetic neurons to the gut. There is viscerofugal fibers that are neurons within the gut projecting processes to outside. Anyway, there's all the combinations, so that's why you have so many of these fibers kind of living or arriving in the gut. So it's pretty interesting that you have a place with so many neurons with such an active immune system. And that's kind of why we are trying to understand what we've been trying to understand in the past several years. So again, we were focusing the first day in the epithelium. And this discussion with João is kind of this change here. See, I mean, this is, but the concentration of microbes distally here, concentration of nutrients here. The second day, and this more early, I was just showing how this compartmentalization of the draining is influencing the immune response. But then that third topic is these neurons that are located in two different regions in the gut, the intrinsic neurons. They are located in the muscularis region. So these are between the longitudinal and the circular muscle. So this is kind of far from the luminal side. So the lumen is here. This is kind of the serosa region, the outside in that image that I was showing. And also there are a lot of neurons in the submucosal plexus here, much less than in the muscularis plexus. And then all the combinations I was saying, neurons that are sensory, communicating with sympathetic chains, with sensory chains, with the CNS. These are the DRGs, the dorsal root ganglia, and so on. There are many potential interactions. So, as I was saying, the question that we are addressing since we started 10 years ago is kind of how can they deal with this chronic inflammation? And then one study that we showed will show that the commensal bacteria, in the same compartmentalization manner actually, the bacteria increases from distal to proximal. From proximal to distal. And the number of neurons also increase from proximal to distal in humans and in mice. And I found that if you modulate the bacteria, for example, give antibiotic or analyzing germ-free or colonizing germ-free with different species of bacteria, you modulate the number of neurons, so germ-free mice, or if you give antibiotics to adult mice, they have less neurons, they lose neurons. And if you colonize germ-free mice with certain bacteria, they gain neurons. So the bacteria controls the number of neurons in the gut as well as the profile. The neurons from the duodenum are very different than the neurons from the colon. But if you make them germ-free, they look similar. So that's the initial study. The second study on this topic was that we found that several enteric pathogens, and we focus on several, but these are non-neurotropic pathogens. And I was talking about this example in the first day. We found that several enteric pathogens induce loss of neurons. Okay, and how did we show that? Again, this discussion of the different regions of the intestine, right? Here is where the nutrients are absorbed. Then you have the distal intestine, small intestine, ileum, and the large intestine. What we did was to give animals salmonella. It's a mutant salmonella that's less pathogenic. and count the number of neurons. We just counted. And what we found is that in the regions that the salmonella is not causing inflammation, like in the proximal intestine, look the number of neurons per square millimeter, 200, 400, 1,000. So it's increasing, right, distally, proximal to this. But if you give salmonella to animals, we observed that they lose about 30% of neurons within days after the infection. And that's not because the salmonella is killing the neurons directly, because it's not neurotropic, but it's very likely a response to the inflammation or to the process that the salmonella is initiating. So this is tissue damage induced by immune response, right? Go ahead.
I have a basic question. This inflammation, it naturally occurs for our own microbiota?
Yes.
Or when we talk about inflammation, it's always something good?
Yes, so the inflammation occurs with our own microbiota. So the gut has a physiological inflammation, which is a level of inflammation. And this inflammation is above that. What is the threshold? We don't know. It's very kind of empiric. But it is correct what you're saying. You know, you have a basal level inflammation. Germ-free mice have less than non-germ-free. Our inflammation is steady state. It's not causing damage. When you have an infection like salmonella that disrupts the epithelial barrier and causes, you know, it changes the composition of the microbiota and it causes additional inflammation. Okay. Yeah.
Can you see this response of the intestine caused by other pathogens that don't affect directly the
Yes,
intestine?
very interesting. That's right. I don't know if I have this data, but since you guys are asking this type of question, I may change this. So you're asking if pathogens that don't cause inflammation within the gut, or within the duodenum or ileum, if they can also lead to loss, right? Yeah. So I'm going to change slightly this. And this is the full presentation. I'm not going to do it because otherwise we don't have time to. But there is a. Okay, so this is the same slide. And then there are 3 projects. I'm going to just show one image for each project. And, you know, otherwise we don't have time. But this I call the first project. When you lose neurons, what happens? And in Brazil, this is what happened. This is the 2014 World Cup. This happened. I was in the stadium, but the majority were people supporting the coup. Okay, in mice, when you lose neurons, what happens? Many things happen. In fact, we don't know what the neurons are doing in every detail. But one of the things that happens is that it changes the peristalsis of the intestine. So what we find is that after salmonella, there is a delay in the transit time. In humans, this is called IBS, irritable bowel syndrome, but irritable bowel syndrome in humans is mostly diarrhea, so accelerated time. But a percentage, 30% is delayed transit. Anyway, there are 2 forms of IBS. What was interesting, and I'm going to get to your question, is that this loss of neurons lasts for a long time, and the functional changes, the delay in the transit motility also lasts for a long time. This is, you know, the salmonella is cleared in 1 week. In 1 week, you don't have more salmonella, but this lasts for a long time. So summarizing what we found was the neurons die after inflammation. It's dependent on the region. They die via, doesn't matter, this is a non-canonical inflammasome pathway. And the macrophages, these are immune cells that are mediating disease tolerance in many other tissues. We found that the macrophages that surround the neurons, you see the macrophages, these are neurons in the gut, and these are macrophages. We found that these macrophages are regulating the loss of neurons. They prevent the loss of neurons. And we showed that in 20 different types of ways. But so the macrophages are around the neurons. We found that the macrophages express high levels of adrenergic receptors. So there is an interplay that's very interesting. You have an infection, not all infections. So for example, the bacteria that we tested, Yersinia and Salmonella, trigger activation of sympathetic neurons located outside the gut. Sympathetic neurons activate, they release norepinephrine. This is kind of conventional neuroscience. You know, I'm not a neuroscience, but you know, it's just basic. They release norepinephrine. Norepinephrine is sensed by these macrophages that express high levels of adrenergic receptor. And then this triggers a program in the macrophages that is neuroprotective. How did we show that? I'm not going to go over this, just to go quicker. So the macrophages express a program that involves expression of this enzyme arginase 1 that mediates the release of polyamines in the environment. And then the polyamines are protecting the neurons from this form of cell death that's called paroptosis. Anyway, that's a very mechanistic, you don't need to worry about this. So we had a second project, which is the Bolsonaro project, which is if you keep infecting, like you're asking, the animals, do you keep losing neurons? So that's why it's called Bolsonaro. Can you reach the number of neurons that's very low? It's triggered our question. See, sometimes there's a benefit. And what we did was exactly what you asked. So we did two experiments here. One is to give a second infection to the animal that was infected previously, to see if the second infection triggered an additional loss of neurons. But there's a problem. If you trigger the second infection from the same pathogen that was the primary infection, you have immunological memory. So the infection is not the same, right? So what we did was to use a different infection. So this is one infection which is infected in the same region. And then we wait for this pathogen to be cleared, and then we come with a second infection, okay? First question is, and that's important for the concept of disease tolerance that we were discussing since the beginning. Did the first infection change the immune response in the second infection? In the sense of pathogen resistance, it didn't, because the ability of the both groups to clear the salmonella, which is called SPI-B, is the same. So in 1 week, both groups cleared in the same way. It was not better, it was not worse, okay? However, the first infection, Yersinia, prevented the change in transit time that was induced by the second infection. So the first infection protected the animals from the second infection, okay? And in terms of numbers, it's very interesting. Both infections trigger some loss of neurons. Yersinia, about 20%, 15%, Salmonella, about 30%. But if you infect with Yersinia first, you don't lose the neurons to Salmonella. So how was this mediated? Again, was via macrophages. It doesn't matter the details here, but if you if you remove the macrophages after the first infection, then the animals lose in the second infection. So the macrophages were indeed protecting. They were modulated in the first infection. They remained there, and in the second infection, they protected from additional loss. If the macrophages cannot sense norepinephrine, then they lose again in the second infection. So the program that was induced in the first infection that was via norepinephrine signaling made the macrophages protective, and they remained there, and they protected via the same mechanism that we are discussing here. Okay, the second, the second is not here. It would be another talk, but the second, which is your question. The second, we ask if a pathogen that is infecting a different region, what happens? And what we did was to infect if this is a helminth, Strongyloides venezuelensis. So this helminth only infects the duodenum. And this helminth does not trigger loss. We never tested bacteria that infects another region to see if it dies there too. But the helminth here does not trigger loss of neurons in the duodenum. It also doesn't trigger loss in the ileum, in the site that normally the salmonella causes. What we found is that if the animals are infected with helminth, which only infects the duodenum, it protects from ileum loss of neurons if the animals are then infected with salmonella. So And we found a mechanism which is here. It's very immunological, doesn't matter. But the way we found is that the helminth is inducing a soluble factor, immune factor, IL-4, IL-13, that circulates and modulates the macrophages located distally to where the first infection was occurring. So that's kind of in the direction of your question. Yeah.
Okay, I have two questions. The first one is more simple. That is, this relationship between the
enteric neural system and the microbiome is on the two directions, meaning that the neurons impact in the microbiota can impact in the neurons, but the neurons can also impact the microbiome.
I mean, the reverse side is less understood. So whether neurons can also impact the microbiota, because it's difficult to manipulate, but there are a few studies that suggest that neuron activity in the gut also can impact the composition of microbiota.
And the second one is related to that. That is, if you think that some type of psychological disorders like anxiety makes this constant activation of the sympathetic system,
Yeah.
then will act in the enteric nervous system, and then can modulate somehow the microbiome, can impact on it.
Yeah, it's very likely. So psychological disorders are certainly changing stuff in the intestine because a lot of them are coupled with gastrointestinal problems. So this is, in humans, is very common. So for sure this is happening. The direction, it probably is mutual. So it's possible that some dysbiosis, like in our case, and I didn't show this data, the animals never recover the number of neurons after an infection. But if you reconstitute, and I mentioned that in the first, if you rescue the microbiota to the original microbiota, then the neuron number comes back to the normal. So that is showing microbiota influencing the neuron. However, the process of gastrointestinal inflammation or gut-brain also will impact motility in the gut, will impact the physiology, and this will impact the composition of the microbiome. So the reverse also occurs. It's much less understood the mechanisms of this gut-brain connection. We have a whole paper on that I'm not going to discuss, but showing how microbiota composition and interplay with the enteric neurons can affect the brain and how the brain can affect the gut, because it's very heavy in neuroscience, but it's a paper we published in 2020. But anyway, I would say both directions are likely happening, and the mechanisms are multiple. So there are, for example, soluble factors that metabolites from microbiota that can circulate and affect the brain. You have cytokines from the immune system, like this example that I just gave here, that can affect neurons distally. Of course, neurons can transmit, and via sensing stuff that's happening in the gut can mediate, and we also showed this occur. Anyway, there are multiple ways of communication, and it's a very rich field now, actually. Okay, let me see. Third, I'm not going to show the third project because it goes very. Okay, I think actually I will stop here because I want to talk about this, the disease tolerance, which is shown here. Do you guys have more questions about this topic? But you understand the concept of disease tolerance here. Just to highlight, I think this slide here is the one that shows most. So this disease tolerance, basically, it's a process that does not affect resistance, does not impact the pathogen load. The pathogen load is going down equally, but affects what people call host fitness. So it's affecting the ability of the organism to deal with the infection itself. And in this case, of course, one result is peristalsis of the intestine, but we measure actually multiple parameters. For example, the ability of absorb nutrients and deal with metabolism in a given amount of diet is also improved if the animals had exposed to helminth before this salmonella infection. So this is showing two mechanisms of disease tolerance. One that is sort of local, it's activation of sympathetic neurons locally release norepinephrine in the gut and affecting locally the macrophages that then protect locally the neurons. versus a distal mechanism that is involved with circulating factors that then impact these macrophages. Okay, so I think we can stop unless you have more discussions. Okay. Yeah?
I have a question. I have a question. Can our body recover this neuronal loss, or they are lost forever?
No, that's kind of what I was saying, right? The neurons, it's a kind of interesting topic. I don't know how to change that. The neurons in steady state are stable. Microbiota change the number of neurons. These infections, what we found that is likely happening is that infection with Salmonella changes the composition of the microbiota. And with this dysbiotic bacteria, you don't have the composition that allows for neuronal regeneration. That was actually the third project that I didn't show. So we are studying how the neurons come back. It's called Lula project. So I didn't present the Lula's project, but it's going to be successful. Yeah. Okay. All right, so now we have like 1 hour, a bit less. Maybe we discuss, because now we covered resistance, the concept of classical tolerance, how you prevent reaction against non-self, and the process of disease tolerance, which is examples from the lab. Of course, there are many examples. I just was presenting what we are doing. The topic that people wanted to discuss is sort of career path. What do you guys want to know about career path? Is there any specific stuff that is helpful for the majority? I was talking to people during lunch about PhD application. The past 2 days we talked about that, but only a few people can go or went to lunch. So what would be the most, the broader way of discussing this? Yeah.
I guess, what are the
You guys can ask, anything, and then we discuss. Yeah.
For these international applications and things like that, what are the gaps that you think people from Brazil or Latin America often don't realize they have?
Yeah, so this is very important because that's exactly what we discussed in the past 2 lunches. So since you didn't go, sorry. I'm kidding. This is the most important component in the application of developing countries. What are the gaps that students from South America or from Asia, Africa will have? Africa much more. We never see application versus a student that is doing undergrad in US and Europe. You ask your question just to to put a framework is what are the gaps in applying for PhD programs in Europe and US, right? So with that in mind, I'm quite familiar with the application process in Europe, but it's very specific to institutions. So I'm going to discuss the US mostly. Gulbenkian, for example, this guy that's coming, Jorge Carneiro, he can talk a lot about that. He's been there since the beginning, so and it's an amazing program, actually. This program that you guys are is based on that program, PGDB. So what I was discussing with them yesterday was that so Rockefeller, for example, it's a different program, but let's imagine that other programs have small variations to that. So Rockefeller receives, most of the programs in the US no longer require the GRE. And we discussed about GRE if you guys want to know, but anyway, it's not required mostly anymore. And that's because it doesn't predict success, even though success is a charged word, but it doesn't predict anything. So it's good and bad. It's good because it doesn't help GRE, and it's bad because when you are evaluating applications, the more kind of exact, and less subjective parameters you have, it could be better, right? If you have parameters that you can measure. But there are no such parameters that are telling if a student is going to be good or bad. So it's a subjective process. And what I was saying was that, what are the components? The components are tests, GRE is gone, so GPA is the only one. What's GPA is your grades in the undergrad or master. It's not essential, but if you have bad grades on undergrad, then is another problem, another little concern that is building in the mind of the reviewer. So if the reviewer has, if the reviewer has let's imagine that there are these parameters here, grades. I don't like grades, but this is a parameter that is some sort of universal, but it's not exact because, A in UFMG might be very different than A in Campinas in biology or whatever, right? Universidade de Ceará or whatever it is. So grades, so you have, before you had GRE and GPA. GRE is gone, so you have GPA. GPA has a concern, which is you don't know if A in a given university means anything, but you know that a D is normally not good. I'm not saying if you have a D, your life is over. I'm just saying if you throughout your undergrad, you don't have good grades, it's one less thing that will help you. Okay? Then you have a personal statement. Personal statement is very important. And it's a long discussion as well. And I know you guys had this discussion in the lunch, so we are repeating a little bit. But the personal statement, I think each university has a different requirement. For Rockefeller has one that I like a lot because it's what is your most significant research experience? That's your personal statement. I think it gives less room for this pre-written personal statement. American way, you go to Google, you write this phrase first, this is the American robotic way of doing stuff. It's of course not good, but at least corrects a little bit of a person that has no clue what to write in a personal statement. And in fact, if you guys need help, it's unlikely I can edit every single personal statement, but I can give advice on that. So just ask me and I can maybe sometimes give an example. I ask the person if it's okay and give an example of a personal statement and so on. But the personal statement you should use, in my recommendation, to tell your trajectory in science. And it's very important that, because you guys, 99%, I think probably all, will be considered minorities in the US, as well as in Europe. And that's a good moment because the people are finally giving attention to minorities. Even though I know that perhaps some of you are less minority than others, of course, needless to say. And this is something that's likely coming later. For example, Latino is minority, but we know that a rich white man from São Paulo is different than an African American from Goiânia, right? So there's differences that are within countries in terms of opportunities are not evaluated yet. But the good thing is that all minorities. So it's important that you tell your trajectory to understand how being a person studying in South America impacted and helped your trajectory in science. So a reviewer will like to see that you have a trajectory that is promising because they are trying to evaluate potential. So the personal statement is that. I don't recommend the personal statement of your grandmother had cancer, then you want to study cancer, because I don't know if it helps. I understand there are personal stories that I'm not making fun of, but it's important to focus on the scientific trajectory rather than what inspired what, unless it's something that you want to talk. But it's very common to say this, and I don't think it helps, in my opinion. It helps to understand what this person is trying to see. And there are no right or wrong. I was talking to two of you guys that had changed undergrad, started again, then doing another undergrad, spend time, this, if you don't explain, it can be bad, right? We were talking yesterday. If you explain, it can benefit you. Well, I did mathematics, and then I realized that I didn't have enough biology to understand biology, so I did biology. That's good. As opposed to no explanation. Why this person has 10 years gap since the first undergrad and applying for a PhD. So it's also an opportunity to explain your life experiences in this context. And I think that's how it should be written, a personal statement. It's a mix between your life experience in the context of scientific questions. And I think it's very important because this is one of the things that will have the biggest gaps in applicants, because this, you can, there is no, that, you know, you maybe we have even an advantage over an American that, you know, I did undergrad in Harvard, I did a master's in MIT, and my parents are very rich, you know. So you may have an advantage in telling your story, right? So it's an opportunity to tell a story because you guys are, by definition, minority. I am minority, you know. And I say I'm not sure I'm a minority because I was not rich, but I was not poor either. So I don't think I'm a minority. I'm white. But by definition, Latino is minority. In any case, it's a good moment for people from Latin America in that sense, I think, to apply. And also for postdocs. If you guys want to do PhDs here, there's a huge decrease in applicants for postdocs in the US. Huge decrease. Like Bernie Sanders says, huge, you know, it's really decreased. PS, so grades, PS. CV, so CV is sort of, it's kind of included here. I don't distinguish much, but yeah.
What are the difference from personal statement and statement?
It's the same, no difference. So some institutions distinguish, for example, a scientific essay versus a personal statement that, you know, so that's why I said that each institution is going to be slightly different. So for example, if say only science, then you don't say your trajectory necessarily, so you have to separate. So again, each institution will be slightly different, but the statement of purpose is kind of the same as a personal statement. CV, there's not much to say. It's just for you guys to check what you put in your CV with people that know. Because sometimes, you know, people just don't know what to write. It's very common to write personal stuff anyway, but I don't have anything to say in the CV. But CV is sort of included a little bit in the grades and in your experience. Letters of reference. I'm going to put letters of reference. So, a rich white American that did everything perfect in his life, you know, got to the best schools, did everything they, you know, I do a summer school in this university, summer school, publishing, whatever. So this person will have a strong GPA because they always thought about GPA. They're going to have a very well-written personal statement because they follow the way of writing. I'm not saying this is all bad. I'm just saying this is the usual stuff, right? Then they have a CV, and then they have letters of reference with people that they know. So you guys are here with a GPA that, as I said, not essential, but it's a way to measure how you did in your school. We all criticize, of course, you know, standardized tests, but that's kind of what they have to measure. The CV and letters of reference, it's a little bit tricky because they will not know who wrote the letter to you in general, right? So what you have to do is to maximize that this letter is meaningful. So how do you do that? First, you know, and again, there's no recipe, but it's very common for letters from Brazil. I know a lot of letters from Brazilians, but I don't know a lot from Colombia, for example. I don't know how people write there, but it's very likely common because it's sort of South America. That the letters are not with content like an American letter is not ideal. American letters are a big problem, and I actually am part of movements to change, to not ask for letters of reference, but then what are you going to measure? It's difficult, right? Anyway, not the discussion. But right now they ask for letters of reference. Every place asks. And so I'm answering this context. Letters of Americans are a problem because they have so many amazing qualities that you have to find less amazing qualities to criticize. So a person said amazing 15 times, and then the other said 10 times. Oh, this said only 10 times. So it's a little bit of a, it's very inflated, the letters. They are very inflated, but people know that. European letters are more honest, for example. They are more like, German letter is very precise as expected, but they can tell a content that is important for the person reading. So one thing that you have to do if you did not do internships anywhere, whatever, and that's fine, outside Brazil or South America, is to make sure your letter is read by someone who knows the type of letters that you're applying to. For example, if you're applying to a European institution, ask someone, you guys just were exposed to 50 people, right? Ask someone to take a look in a letter that the person is, not necessarily you, but we were also discussing yesterday, you may want to write something for the person to help about your trajectory that you want to include in the letter. I'm not saying that you have to write the letter on the person's behalf, but it's always helpful if a person doesn't have time, that you give something. I don't think there's any problem with that. The other topic that is also very complicated is, I'm going to give an example. We went to lunch, and can you write a letter for me? Because I'm applying to Rockefeller. And my answer is the following. In general, if the interaction is somewhat meaningful, let's say we spent several hours talking and I had a good impression, and I can say something that is constructive, that's okay, because if you're applying to the place that I work, I at least know the person. Of course, it's better than not knowing. But this is not black and white. For example, you don't write to a person that you just wrote an email and ask for the person to write without any interaction, or a person that you just ask a question, because if it's not a genuine interaction, the letter is not going to be meaningful, and then it doesn't help. So in this case, you may want to say, I'm applying to your program, just keep an eye, or if you need to talk to me or whatever, I'm happy to chat about science. So try to make an interaction in this case that is a bit more meaningful. It's no doubt that this type of interactions can be important, but also I want to avoid the American connection that is superficial, because then I don't think it helps. So the letter has to be meaningful. I think you were asking yesterday if you have the, who asked this question? I forgot. You asked, is a letter one paragraph? It's difficult to know. Of course, there's the John Nash example. He's a mathematical genius. That's a good letter. But I would say a letter from a person without any knowledge of you, even if the person is famous, it's less helpful than a letter that is well written of a person that knows you and is not famous, in my opinion. Again, it's not black or white. There is gradient here. So letters of reference are very important, because in the end, you're going to have, in general, you're going to have university that may not be well known, even if it's the most famous university in South America, or among the most famous. They know, but they may not know if the department is good. You have to assume that. So you have a little bit of disadvantage here. The letters of reference are complicated because the letters of reference will be from people that are not very well known. So that's complicated. So a little bit of disadvantage here. So you have to maximize on that to increase your chances. And definitely, you guys are in one of the most selective programs, right? You had 6 months of strong kind of interactions with people that are among the leaders in the fields, right? In many fields. So just by being in this course, it's an advantage for you. right? And you may want to have a contact, and I'm not saying, I'm sorry for the other people, but you may want to engage in some people that you're applying for some universities that they belong and ask, you know, it's okay for, what do you suggest? I think it's part of the, I was telling Natalia, it's part of this program objective to do that. So don't feel shy to do that. It's fine, you know, and contact people. You want to apply to a specific department, the person gave lecture. I mean, of course you should contact, right? Everybody that I know that gave classes here, I think you guys are a very strong group of students, so you are in an advantage over the rest of the people, right? And this can be within the letters of reference aspect as well as a personal statement. So anyway, I think I covered the basics. Any question on this? Paper. So I guessed, okay. Do you need to have papers? Of course you don't need to have papers. First, because papers published in the undergrad, generally, you are in the middle of the authors. Very rare you have a first author paper, because it's not supposed, you guys are doing classes, you're not supposed to be publishing papers. If I have papers, a problem? Of course not a problem, it's good. You know, but I don't want you guys to go with the mindset that a paper is the objective at this stage in the career. It's not. It's much more relevant that you have meaningful research experiences, interactions, that you are active in your trajectory, and that is meaningful to you. It's not superficial. It's not something you're writing just for to check the mark, right? And I think this is much is better perceived than publication. I don't care about publications when I'm reviewing, honestly. You were asking, oh, if, you know, what if I have? Well, it's good. But don't, you know, we discussed this for a long time, but. Okay, I didn't answer. Paper.
Yeah, just regarding the papers.
Yeah.
Here in Brazil, in the PhD programs, postdoctoral programs,
Yeah.
usually they use a rank, and you have more points if your paper it's published in a qualis A1 or
Yeah.
A2. In US, they evaluate by some metric like SIGT score. They
No,
use
there's
this
no
or-
such score. Yeah, I don't know any of these scores there. But people know sort of. I mean, the publication is another 2 hours discussion because it's also a broken system, right? So, yeah, I don't think there's an easy solution for that. But people just know. Sometimes this kind of a specialized journal that people know is a good specialty journal. Anyway, I don't think there's a. Of course. There are also the journals that people never heard of. So these are less meaningful, but I don't know, you know, A1, AB, or whatever. Especially at this stage, I mean, it doesn't really matter, I think. I don't know if Annelise was asking, you're asking a specific level as well when you were discussing, you know? A specific journal subtypes, like, is that was in your mind as well, no? Okay.
The question I have is, I thought of it when you mentioned in one of the previous classes, your evaluation of how a change is due to occur regarding whether people who are doing analysis and data work should become first authors and the like.
Uh-huh.
So, and based on that...
British accent.
I'm Brazilian.
Okay.
I'm from Rio.
Okay.
Close to João.
Okay.
And
So you're asking about the role of computational biologists in the authorship, yeah.
But also regarding what you refer to now as Anne Nielsen's case,
Yeah.
like he did a
Mathematics.
master's in mathematics in
Yeah.
went away to do a biotechnology degree. I tend to think about those unusual trajectories a lot because ever since the beginning of my rather sparse range of interests,
Yeah.
and there was no clear, there isn't already even yet a clear path
Yeah.
for I should follow. How usually it is seen, those unusual trajectories? Like I, for example, did physics and biomolecular science in the undergraduate, then a pure master's degree, pure mathematics degree. So I sometimes wonder if that would be badly seen because.
Yeah. So I would say that because unusual includes so many things that I can't answer, right? Unusual includes good unusual, bad unusual. I don't know if there's a bad unusual, but there are, a person, just decided to quit science for 20 years and wants to go back to the lab. I mean, that's in a way bad unusual because a person that has no contact with lab for 20 years, I'm giving an extreme example so I don't hurt anyone. But I would say the opposite, honestly. And I'm going to have two answers. If you're applying for, like you're saying, like if you're applying for a toxin specific, I study mechanisms of toxins from snakes, whatever, or from, scorpions. And I study the mechanisms of action of toxins, and that's what I want to do forever. So in that case, maybe an unusual, a mixed background maybe is less positive than a trajectory that was studying toxin biochemistry the whole life. In general, in my view, the type of, the Nielsen and others here that have done, apparently yours, that is diverse and multidisciplinary, is a plus, in my view. I know many examples. I mean, Gabriel, one of my best friends that is in Rockefeller, is a musician. He didn't do undergrad, only in music, actually. And he did just masters then in immunology, then he went to the US. So he is a piano player, and he was a piano player until 25 years old. Okay, so I don't think he didn't get into Rockefeller program, he got at NYU. And I think it was essential in his case that people know who what he's trying to do. In many cases, it's unclear what would have happened if he didn't have contacts via his masters at NYU, right? Because who is this musician, what he's going to do? So what I was telling the Nielsen was, in your personal statement, it's very important that you address very well any gaps, any change in direction that you had, because that's what people want to know. I had a student, Paul Miller, who published a lot of these papers here. Paul published, I don't want to count because it doesn't matter, but he got the Weintraub Award, which is the best PhD in the US. So Paul didn't get at the Rockefeller program initially because of his low GPA, and he was engineering. He was doing engineering at Stony Brook, and he had a low GPA. And I said, this guy decided to quit engineering and switch to biology. So I argued, because I knew him, that he would have a chance, in doing the PhD. And then he got in after a discussion with the committee and so on. In his case, was also a gap of years without research. So it was a lot of red flags: low GPA, engineering, switching, and a gap. So in this case, either a person, and I said, Paul, your personal statement did not address from the places I know and the people I know, and that is, of course, is a limited sample, that it's a positive thing. It's very rare that changing direction is going to impact you negatively. But if it's not addressed, it sounds like a red flag. It's like what was happening. People want to know that. So this, you have the space in general to address this, and you should address without any problem. I mean, this is, and I was telling to the news, this can actually be a plus to you. You can say, well, that's very interesting. And that's what you should try to make sure that is clear, you know. And regarding authorship, that's a difficult topic, and I think the field has to change as a whole for incorporating more the computational biologists as drivers of projects rather than helpers of projects. But it's going to happen, it's for sure. I mean, even at Rockefeller selection, we had 2 of people that were coming from computational biology labs that were offered jobs, and it's kind of the most disputed, kind of the highest demand type of job, and I think the transition for within the lab, it's going to take some time, but it's going to happen, that they, you know, people will be first authors, they will have a career path that is similar to anyone else, and even, you know, have even more opportunities. So I don't think this is going to be, it's still not there. It's, if you look in general, it's not what's happening. But also you have to have a balance, right? If you just analyze the RNA-seq of 10 people, it shouldn't be 10 first author papers. So how this is going to be done also has to be discussed. Okay, more questions? Yeah. I think it's not, I have to repeat.
Yeah, it's just a very quick pointed question. So every program in the US now, they usually have like some kind of diversity form.
Yeah.
And there is always one question that I think they are forced to report to the US federal government, which is usually like a long thing that you have to do, which is like, are you a Hispanic or Latino or otherwise Spanish speaking?
Yeah.
Are Brazilians included in that question?
Yes.
Oh, okay. I never found this information. Okay.
Yeah, the definition that you can look that people use both is the NIH definition and the University of California definition, and they have a specific, and that includes Brazilians. So yeah, you normally just, for example, your case. I mean, I'm looking at you would be a white but Latino, right? Like I am a white and Latino. So, but Latino as a whole are included already in the URMs. But it's not considered people of color, as it shouldn't be, right? And necessarily, right? You know. No, I think this is a big gap. I actually talked to my colleagues there because I said, you know, I think a Chinese person coming from rural China that has no science and went to Shanghai and went to the, you know, Tsinghua Institute of Immunology, or went to Shanghai University, and is not considered a minority, but you know, whatever, white rich guy from São Paulo that did Mackenzie, USP, whatever, and it's considered a minority. It's weird, honestly. So, but it's a problem that should be addressed after you address entirely the URM project problem first, which they are, I think, doing now. And then you say, well, let's look more carefully. And in fact, at Rockefeller, my friend Soheil and I organized the first diversity symposium for early stage postdocs. And we took into consideration that. So we select exactly a Chinese woman from rural China and Egyptian. You know, Egyptian, Northern Africa, is not considered URM. By definition. And I said, well, this person told a story that makes sense to be URM. So as the same as, of course, a person that sometimes it's a white man, American, but living in a rural US that had difficult you know, was a poor family. So, you know, the more granularity is, I think it's crucial, but it's going to happen, I think. Underrepresented minority, yeah. Sorry. Yeah, is underrepresented minority is the main term, and then you have POC, people of color, which is the other term.
So I have a question regarding the choice of the advisor. Usually in these PhD applications, postdoctoral application, have a short period to look at your research project, talk with your possible advisor,
You're
And
talking about which stage?
when you are applying to a
But that's the thing. In Europe, QBanking, for example, you apply for a lab. In Rockefeller, in the US, you apply for a program. You never apply for a lab. There's no that I know that you apply for a lab.
okay.
In fact, I have in my computer a standard reply. You should not apply for a lab. You should apply. So I received so many people applying to my lab. I said, "Oh, you can't apply to my lab." You have. But it's important because it's not maybe obvious because if you've never been there, you know. But it's very big difference in Europe. I would say in Asia. I think it's varied actually in China versus Korea, Japan, but well, this part of Asia. But in US, 99%, I would say, it's going to be a program, not lab. And in Europe, the opposite, probably. A few exceptions,
Yes.
like the Crick Institute, I think is general. Crick was created, inspired by Rockefeller, it's a very similar program. There are QBanking, I think, QBank actually, I think QBank is a program. You have rotation. So there are a few places that kind of do like the US, rotations. Anyway, so I interrupted you, but that's an
Yeah.
important distinction.
Okay, so I was thinking about how to make a correct choice when you're choosing the advisor, because you're gonna have just few moments to talk with them.
Well, that's
and-
why I don't think you should choose. I actually think in this example, the US system is better than the European and Brazilian systems.
Yeah.
And I don't know, that's why the rotations are important. Your rotation, you spend 3 months in the lab, you know better the person knows you, you know. I don't accept every rotation student. And not, you know, rotation students don't accept my lab also, so it is mutual. And I think that's very good. But what to do in Europe or in Brazil, how to choose? My recommendation is to spend most time possible. So what you gave example, I wouldn't do. Because how do we do interviews in the US? That's also very good. Huh? No, the example of just a conversation and you make a decision, right? So I think for this criteria here, if I had one of you in my lab for 2 months, it's the main criteria. No comparison with any of this. I don't care about the personal statement, your CV, your grades, if I know you, right? Unfortunately, there are 1,500 people applying to Rockefeller, and we can't do that. But anything that is closer to that is better. I know better some of you and someone that never came here, and so on. I'm giving an example. So if you can spend time with the lab, good. If you can talk to the mentees, to the trainees, good, right? Don't talk to the PI only, talk to the person. Is it supportive? Does he spend time with you? Is a supportive environment or is a toxic environment? Right? In Brazil, unfortunately, it's still very toxic for a woman. In South America, I mean, I know so many cases when I was a student, it's like harassment. This cannot exist. It should be zero tolerance. And if the lab does that, you don't go to this lab. And you don't know that by talking to the PI. You know that talking to the people, to the trainees, to the technicians. Talk to the person cleaning the cages. Is this person treating you well? And I think all this is important. And it's not a discussion that is here yet, but it should be. Yeah.
I have a question regarding the number of letters of reference because I'm always, confused about it. I don't know if it's very good to have a lot of them, but then in the other perspective, it could maybe make the person that is evaluating it think, no, why there's...
I never thought about this deeply. I know that there are many cases. So the question is a number of letters of reference. I definitely don't think you need more than 3 or, maybe 4. You don't need more than that. And you should pick the best to become these 3 or 4. But I understand that there are examples, and that's very common in Brazil, that a person spent the undergrad, the master's, the PhD, whatever, whole career in a single lab. And then there is one person that can write properly this letter of reference. So in this case, I think it's very strongly recommended. So you never had experience outside a single lab. So, but you had a good experience in this single lab, right? So it's also good, was also good. So I think in this case, it's very important that you talk to a collaborator of the lab, a senior person, better than a person from within the lab. So you go to this, scientific collaborator, priority. If you spend a lab 5 years, someone else should know you, right? Ask this person to write, not the PhD student or the postdoc in the lab. Then academic, people that gave classes, very common. In every single applicant I have the teacher of a class giving. This was the best. It's not something that it counts as much as research, but it's important academic performance. So a teacher that you interacted for 6 months that's another person to write. And then you go down in the stuff. The left I would ask is a person from within the same lab, even if the person has an important position, because that almost doesn't count. Yeah, and I would say, I think it's important to try to have 3. Even if you have only 1 to write, then you find a course, a person that gave a course to you, and some collaborator.
With all of the recommenders, if for example I have one recommender that she never was like my teacher, my professor, but I was teaching assistant for her. Is that a good recommendation letter?
In a course?
Yes.
That's good. That's the one example I gave, right? It's a person that in academic interaction, not research. That's very common, and it's good because this person can write well, properly about you. Other questions or topics? Okay.
And I was wondering if, for example, you don't have that much research experience, how some extracurriculars that you do count for your application? Because I know in the US it counts a lot for undergrad, but for PhD programs, how it is seen if you are part of groups and of students, things like that?
Extracurricular activities. So let's say, in general, in the US, if you did undergrad without a lab experience, it's uncommon. Almost 100% of the case, you're going to do what they call post-bac, which is the master's here in practice. It's a technician work, but here people do that as a master in a way, in my view. So I think this is almost essential if you're applying to the main programs. If you had no lab experience, that you shadow a physician or that you organized whatever, that is not going to help a lot. So almost nothing can match some time of research experience, even if it's 6 months, right? So I would not recommend counting on extracurricular activities if you don't have lab experience. Then you should have lab experience. If you're applying to a place that is wet lab, of course, that can be different if it's a bioinformatics and then you have some bioinformatics experience. That's different, right? Is that answering your, or you have a more specific?
No, it's just because I'm from applied mathematics, so I don't know if you know more about this.
You don't need wet lab experience.
Okay.
No, that's for sure. No. I meant lab experience, but can be dry lab. That's normal.
I mean experience.
We just accepted someone, Victor Ramos, Brazilian student at Rockefeller. He just went to rotation in my lab. He never held a pipette. It was a bit, oh God.
I'm talking more about research experience in general, because from the major that I did, it was actually very uncommon to be to do research as an undergrad. I had professors telling me that we were not supposed to do that as undergrad because we don't know enough mathematics to do research, which is can be debatable, but
No. Yeah, no. I don't think you need to have wet lab. Of course, if the program that you are applying, and so I'm saying in general. In general, person like without any wet lab experience will preferentially apply to a bioinformatics PhD program. In general. But there are many PhD programs that are broad, and many others that Rockefeller is abroad, so it doesn't matter, right, the field. But there are many others that they say we are also interested in people with dry lab, so it would be program by program type of, is this the right program for you to apply? It's a question that everyone should ask first, right? But what I'm trying to say is that it's very common people with bioinformatics only experience, and that's fine. No problem. But you should select the right fit for you. And the right fit, it can be many things.
Using this answer that you just gave us, what do you think could be good kind of points for us to consider when we are choosing the programs? And once you've been accepted to, I don't know, we apply for 4 programs, we've been, I don't know, accept for all of them. What are the things that you can make, then help you make a decision?
Well, so there are 2 things. One is, if you see yourself as a PhD student, how do you see yourself? Do you see yourself, I'm going to give specific example. I see myself in an institution that has a structured PhD program, which is a lot of courses, mandatory, a year and a half of courses, that you need to do 3 or 4 rotations, that everything is planned. That is, for example, MIT. Biology is like that, structured PhD. Rockefeller PhD program is unstructured. Nothing is mandatory. So if you apply to Rockefeller and say, I want to apply to Rockefeller because I like structure, that's the wrong thing to say. It's an extreme example. I'm just saying this. If you apply to Harvard Neuroscience, and Harvard Neuroscience has a tradition of not accepting people without wet lab. That's not the right place to apply, but I'm not saying that's the case. You have to study a little bit the program, asking, and it's okay to write to the dean's office to ask, people do that, and they have to write back to you. They will write. At least I know that they write back. At least at Rockefeller, they always reply. So they say, I have this trajectory, I'm the right fit, or whatever. They should say, well, I think you should apply to this program. So what I'm trying to say is that is the mutual thing you have to look. When you see yourself, do you see yourself in this program? Let's say you don't want to do wet lab research. Then you have to maybe find a PhD program that is open to that. And there are many. Rockefeller, for example, has a tri-I bioinformatics program only. But people apply to either. I don't know, they apply. This guy, Victor, applied to the bioinformatics PhD program and to the general one. And he was accepted in both. I think you're looking to me and say, you did not answer my question. But I just cannot answer a question precisely because that will depend. So what I'm trying to say is, do your homework, look at the programs that you're trying to apply, and see if you see yourself in this program is a good program. Let's say someone wants to work with mathematics applied to immunology. I'm not saying that that's impossible at Rockefeller because Gabriel does that, for example. But let's say Gabriel doesn't do that, and there are 4 immunologists at Rockefeller. And you say, I want to study mathematics, immunology. There is no single person doing what you're proposing. That's difficult because the person reviewing your program said which lab she's going to work, right? So these are examples that are obvious, I know, but it's just to kind of tell you things that you should consider when you're applying. And always contact the dean's office of the institution and ask if you fit a criteria that they can't tell you if you're going to be accepted, but you say, is this type of trajectory fine with you guys? And, I don't know if I made you guys more relaxed or more nervous. What I'm saying is that it's very interesting that Brazil is a place, and South America, maybe Argentina is a bit better in that sense actually, but that has less people applying for full PhD programs there. It's very, very few people, and I don't know why actually. I think the main reason is because Brazil is very common for people to stay in the same lab and do and continuous, like just inertia. And then it's also very common to do a sandwich, right? So people just say, I do a sandwich, right? But I think, and I'm not here to try to make commercial, propaganda for applying food there, but it's definitely something to consider. It's a different experience, and it's if you can spend more time in a place right now with more resources, in general, is good. And I'm not trying to destroy Brazilian science. It's just I'm trying to improve, but it's your decision in the end.
I know we have only 3 minutes, so I don't want to take any more time.
I'm fine. Yeah.
But could you comment quickly about funding? Because I know a lot of PhD programs, not all are fully funded. So for us Brazilians, it's impossible to go there.
Well, if in general, and there would be very rare exceptions, if you're accepted to the PhD program, you don't have to pay stipend, and you're going to receive a stipend. In biology, biomedicine, bioinformatics, ecology, all this, for what I know.
Okay.
I know that in humane science, that's different. You do MBA or whatever, that's different. But in the fields that you guys described here, I'm pretty sure that the vast, I don't know, I don't want to say 99%, but the vast majority of the programs will support. You don't have to pay monthly fee, and you're going to receive, and normally you're going to have subsidized housing. Then, within that, there are differences. And it sounds that I'm doing a commercial for Rockefeller, but Rockefeller, for example, you don't have to, you not only receive a stipend, but your lab that chooses you also don't have to pay for your fellowship. It's paid by the university. In general, the vast majority of the programs, the lab that hosts you has to pay your salary.
Okay. I'm from ecology, and I've seen some programs that they aren't fully funded, and they talk to the professors from the past few weeks. What they say is that if you come with funding, if you already had some kind of fellow and you bring to them, you're more likely to be accepted, especially because in ecology, you choose your lab, like in Europe. So I don't know, I was kind of in doubt
ecology,
about
I guess,
this.
because there is less federal funding for ecology than biomedicine. It's possible that the PhD programs are less supported. But I would doubt that they require that.
But it's...
It's better,
It's better.
But a person coming from South America, they unlikely will say, well, why don't you So I would apply. But what I'm saying is that if you are accepted, even in ecology, and I could be wrong, maybe you say you're wrong, and then I change. Even in ecology, if you are accepted, you're going to be covered. They say, well, if you have funding, it's easier for us to accept. That's weird because it's kind of, it's a bribery in a way.
But I've heard that.
Rockefeller has a funny story that one of the donors, these billionaires that donate, they wrote a letter to the dean and said, my grandson is applying to Rockefeller. I would be very glad if you guys accept him. And then Rockefeller wrote a letter back with a check of everything this donor has given the university and said, well, this is to be decided by the dean's office. And they returned the money that he had given in the history. Anyway, it was one case, but I find weird that the decision can somehow be influenced by the fact that it should be these things here, right? Not, I don't have one here like this. But again, contact the dean's office, and I would say, as a principle, it's weird if the federal government is sponsoring a PhD program in the US. That's weird. I don't accept any fellowship from Brazilians anymore. Even if they have, I said, I don't, you decline and I pay because, the funding is 1,000 times higher. It doesn't make any sense. Still, should Brazil have fund? Yes, for people to live. But I think people there, well, they should have, because sometimes helps for a person to live, and that's beneficial. But the people outside should be a bit more conscious that the funding here is zero, right? So,
Okay.
yeah. Okay, one more? No. Okay, there are two more. Ah, yeah, we're gonna take
Do you want to
pictures.
ask it? Yeah. So yeah, in many programs there is, after this initial selection, they invite you for interviews.
In general, yes. In general, they
Yes.
are doing Zoom now because
Uh-huh.
they learned with the, yeah. But yeah.
Yeah. So do you, is this like, are there any comments?
Tips for the
yeah,
in-person-
comments
yeah.
you can give about that?
The interview is very interesting because the interview allows for the selection committee to be a bit broader. This person has a weird path, but it's interesting. Let's see what the person. So this interview is important, and I know that it's also tricky because people deal differently in social interactions, right? Carolina, I don't know if you noticed, she's not easy in social interaction. She has, she's shy, and but some people are very extrovert, some people are introvert, and that is kind of affects. But anyway, the interview, what you're trying to assess is if the person wrote a Google statement, or if the person meant what is written there. It's nothing special, but it's very important that you engage with the person interviewing. For example, Rockefeller used to accept in paper prior to the interview. Then the people who would come to the interview accepted. So it was very weird because when I joined there, it was like that. And me and my friends that were junior, we were like, it's kind of weird. I'm talking to the interviewer, to the student, and I'm trying to convince that he's not making any effort to talk to me. So we changed that after second year, and now you're not accepted until the interview. But we accept 80% of the people that we are interviewing, right? And I would say, in general, it's kind of high percentage. I would guess at least 30, 50% will be accepted because it's very expensive to organize the whole interview. So they will, once you go to interview, what I like to say is you're taking out the kind of crazy nuts, like that shouldn't be there, that are, crazy should be there, but crazy in the sense that the person is not, doesn't know anything about what did, and it's also taking people that are just maybe not what they wrote in paper. So it kind of is trying to say, is this person, should I, this person be a student? So you don't have to perform. You try to be yourself, because you can be yourself as an introvert, there are different types of, but it's very important to engage a little bit if you can. To show, instead of say the phrase that is kind of memorized, to kind of show flexibility in discussing the topic. Normally what you're going to do is that they're going to tell you about your research experience. And then you're going to say, oh, I did that, and then I went for this, didn't work. And then they say, why are you interested in our program? Yeah, I thought your program, and so on. So there's nothing special. But it's important to show engagement. That's kind of, for me, it's kind of thing that I try to look. Yeah. Okay.
No, I just want to thank you very much for coming here and teaching the students. Yeah, let's take the picture.
So you guys took a picture with every